So it is my absolute pleasure to introduce Joaquín Navajas Acedo for the 2025 Society for Developmental Biology Trainee Science Communication Award. I cannot think of anybody that is more deserving. So I have just a few slides to introduce him. So Joaquín is extremely online, as he has said himself. So there was a wealth of information to find about him. Now, this is a YouTube video that you see behind me that is outlining his education, and he did undergraduate studies. Can you see it? Yeah. He did his undergraduate studies in Spain, and then he went to the United States, and so you can see the pictures of who he trained with here, and there's Tatiana Petrovsky. And then he is now a postdoc in Switzerland in Basel with Alex Schier. There's a picture of him online, graduating, getting his PhD. And when I was wanting to get a little bit more information about him, I mean, there just was so much. I learned here that he loves reading, watching movies, and going to concerts with his wife. The one thing I couldn't find is the picture of his new baby, and I was desperate for that. But I want to just give you guys a little bit of why he became this nominee. So I met him as Mad Scientist on Twitter, and he is extremely popular. So if you look at the number of followers, that's how it's judged on social media. That's when he was on Twitter, but now he's locked it, and I think he's gotten rid of most of it. That was where I was hoping to get the baby pictures. But then when Twitter sort of changed, and I refused to call it anything else, he moved to Blue Sky, where he immediately got just as popular, so you can see his followers there. And there he has really just been somebody to bring our community together and to tweet about science and retweet people's papers. And I mean, it is just a fantastic. I grew to love him before I ever spoke a word to him. But then at some point, I asked him whether or not he would like to man the SDB Twitter science feed to be able to help us, relay all that information, which he did. And he immediately just shot the Twitter for SDB up to many, many thousands of followers. And that job he is now stepping down from, and Martin Esterman is going to do just as great of a job. But so thank you for your service in that SDB manning of the Twitter feed. So why is he so popular other than just having great ideas? He's hilarious. And so that is him there. There's a cat blowing in a tuba, and he says, after 3 years, I think I've got a phenotype. Here's another one I loved. I'm putting this on my CV, be right back, BRB. There's an important account that followed him, so he's like super excited. And there he is just kind of telling us that he's feeding the fish. So, I mean, these are just small examples of the constant hilarity. But, what I think is so important about Joaquín is that he's just so relatable. He's honest, and he says things like this here. He just picks up this tweet from somebody else that says, I hate literally every step in the filmmaking process. The only thing I hate more is making a film, then making a film is not making a film. So somebody else said that, and he picks it up and he says, writing a manuscript. And I'm like, yes, that is exactly right. He understands, and he always addresses everything with humor. Here's another one. Casual small talk, me, disturbing fact that nobody asked for. Relatable, completely relatable. He shares a lot, so I learned that he has tattoos, so now so do all of you, because it's everywhere. There's his tattoo, very cool, by the way. And he shares his moods, which we all have. There it is, current mood, and he's having a hard time, I think, writing a paper or something. So, but, beyond all the funniness and the amazing science communication, he's just an amazing person. And here's an example of an email that he sent to me, Ida, and Marcia, and I know it's personal, but I'm just going to share it anyway. He says, hi, Ida and Marcia and CC'd interested parties. Would it be possible to consider adding to the guidelines of the meetings to please, please, please not use red-green, or more generally, be more colorblind friendly in the figures shown at the meeting, or at least bring it up at the BOD meeting? And so this is the kind of thing he does. He's not colorblind, but he cares about colorblind people. He's extremely inclusive and just about the best science communicator I can think of. So congratulations, Joaquín.
Hello, Puerto Rico. I'm very, very happy to be here. You have no idea how happy I am to be surrounded by my people, mi gente, the developmental biologists. And today I'm going to talk about two things that I consider important to me. One is what I do, and the other one is the developmental biology community. So, thank you, thank you for nominating me and giving me this award. And I hope that I can live up to the to the hype. So I work in zebrafish, and I use it to understand what I think is a fundamental problem, which is how our neurons make different. If you think about a neuron, you think about this. Some neurons are big, some neurons are small, some neurons have a lot of dendrites, some don't. Molecularly, they express a different combination of things that serve different functions. And how the nervous system generates neurons is, molecular and cellularly is not completely understood, right? So what I'm trying to do during my postdoc is use a model to understand this problem. And I use these neurons in the dorsal part of the spinal cord called Rohan Beer neurons because they got described independently by Rohan and by Beer. They actually got described by Sigmund Freud by 10 years. He described them in lampreys, actually. So there's a fun trivia for you. And I study them because there's only a handful of them. They're very easy to image and to manipulate. They develop very fast, and they have a physiological relevance. The thing with these neurons is that for 150 years, people thought that they disappeared. So they formed, they underwent pleural cell death, and then they got replaced by the DRG. But sort of by accident, actually, I found that this is not true, and they remain. And I published this together with Kusena's lab, and I was very happy because this was a solo author paper, corresponding author, my very first paper, and I was very happy that I could publish it in Developmental Biology. And let me do an intermission here and tell you that publish often, publish well, publish in Developmental Biology, the official journal of the Society for Developmental Biology. There are many perks. So you're a member of the society. They guarantee that they will send you out for review, and they do publish developmental biology without shame. They don't rebrand it as something else. They call it developmental biology, which I think is a very important thing to do. So, going back to Rohan Beer neurons, I study them because, like the DRG, they serve a physiological function. So they take touch, temperature, perception, nociception. They do this because they express specific ion channels and signaling molecules. And when I started this project, it wasn't clear what these were. So just to summarize, many years of work in this slide, I found that they are complex and heterogeneous at different levels. So the first level is the transcriptome. The second is axial level heterogeneity. So some of them are in the anterior part of the trunk, in the middle of the posterior. And they are also heterogeneous even between individuals, which is, suggests that there's some sort of stochasticity to the mix, which I like very much. But this was done one gene at a time. If you've ever done an in situ, you know how painful it is. So I decided to take a more high throughput approach, and instead of doing one gene at a time, I did many, many genes at a time. So I teamed up with another postdoc in the laboratory, Yinan, and we developed this custom spatial transcriptomics that does not require sectioning, so it's whole mount, like, this entire trunk was 150 microns, single molecule resolution. You can see how it looks like, so you can quantify it. So if you do this for raw neurons and the markers that I selected from a single cell, you can do as many runs as you want. And then, how does this summarize into, the distribution that I mentioned? And basically, this cartoon shows that what I found is that there are these different layers actually translate into the different subtypes, and how they develop in space and in time. So there are channels that are everywhere. There are channels that are mutually exclusive, and then within these two, they are subtypes. But like the other thing is this at the molecular level, but how does this happen at the cellular level? So through other people's work, we know that rhomboidians are born in the neural plate. They actually induce the other less known cell type in the neural plate called the neocrest cells, and they they born there, and people know this based on BRDU pulses, but they didn't know the lineage. So I image using light sheet, and then what I found is that their lineages are extremely simple. So either I see them dividing or I don't. So that means that I can do mapping of transcription factors and processes with high resolution during the development of these rhomboidians. But if I take this, what I did then is take the video, fix the animal, and then I've done spatial transcriptomics on it, because now I can correlate lineage and molecular heterogeneity. And just to give you a teaser of it, on the top is the islet signal from the light sheet. In the middle is the segfish, and then the two mutually exclusive neotrophins that they express. And so far, I haven't found any proof that these related with at least cell division, but who knows, stay tuned. But I think that these rhomboidians are a very cool model to understand the process of generation of neuron diversity at the different levels, molecularly, cellularly, but also things like morphology or how the circuit assembles. They also have very interesting properties during evo vivo. Amphioxus have them, lampreys have them, zebrafish and frogs have them, and then trying to figure out whether drier vertebrates may have them. So that is the rhomboidians. So very, very summarized. But now let's talk about this other guy, which is why we're here. So, like Ondine said, I'm extremely online, and I use it to learn about other people and to promote my stuff and learn interesting things. So when I'm the living proof that when your advisor says that you spend too much time on social media, no, you don't. Yes, you do. But I think that, like I said, the other passion that I have is developmental biology. And I drew this for when Alejandro Sanchez Alvarado got elected to the National Academy like 12 years ago, 13 years ago. But I do think that we should all very proud to call ourselves developmental biologists, and it's something that we should take with us, wherever we go, with a lot of pride. Like Ondine said, I manage, several social media accounts. Mine, the development of for the Society for Developmental Biology, and another one that is zebrafish. Focus is started by Kevin Thyssen, who actually got this award 2 years ago, and then Michaela, Charlie, and Maddie. So I am also part of the PCC, which is all these people, which are wonderful people, and every time we meet online. It's very, very fun because you get to see, how much they care about the mission of the society and what they can do to bring the society to not only the constituency, but also the general public. I'm very grateful. And, even, this year I'm stepping down and, Martin is going to be my successor. And he's wonderful. You'll see, he's very motivated and very hopeful for what he's going to do. So, changing gears, when I think we should all be talking about today is this, which is that science is under attack. And this is no hyperbole, this is true. And there are many, many examples on developmental biology, stem cell biology, cell biology, our field, other fields, right? And over 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 again, science is under attack. The funding is being cut. And, science, basically, we are in a situation in which our line of job is under threat. So what do we do, with all of this? So if you think that I'm exaggerating, and I don't think I'm exaggerating. So just in the United States, this is data from just the US, but all the science budget is being cut. Not only the NIH, but the NSF. And I think that this is not okay. But also what accompanies this, which is, I took this term from business, which is enshittification, which is that you take a product that was good and then you make it bad on purpose, right? One example is now they put in AI and everything. But I think that it's important that we acknowledge that there is a deterioration of the institutions by electing people that does not believe in science. And these people actually quote, for instance, this paper or whatever you want to call it, which is the way of deteriorating the literature, because this thing is indexed in PubMed, right? So this is how we lose our line of job. And I think that, we need to acknowledge and, push back. So how are we as embryologists, stem cell biologists, cell biologists, biochemists, molecular biologists, zebrafish, mouse, chicken, flies, beetles, smoothworms, spiky worms, jellyfish, lizards, octopus, developmental biologists, everybody, going to confront and resist and show up for each other now that we need each other the most? So I think that it's important that we do not give them an inch. And this is important. We do not capitulate and we do not speak in code, right? Because any person studying gastrulation can tell you that there is a power in diversity and in numbers. So we have to do this together. So how do we do this? We do what we can, whenever we can, if you can. So, talk to other people and get ideas. Please tell somebody that you think that can do something about this. Tell them how important it is. Participate and organize protests. And stay informed, right? So don't not only what is in the media, but what is beyond what the media are telling you, and inform other people. Tell them that you are a developmental biologist. Tell them why it's important what you do. Tell your neighbor, tell your family, tell your mailman why you think it's important. For instance, borrowing this from John, tell them that developmental biology is important, because people don't grasp what developmental biology is. But if you tell them that birth defects are the number one cause of death in the first year of life here in the US, and that is a developmental biology problem, then they will understand. Tell them that diversity is important in developmental biology the same way that you need many, many cell types to function. Diversity in science is important. And tell them that DEI initiatives are crucial, and it's important that we support this. Tell them that science is a very good investment. So if they care only about money, tell them that for every dollar that they put in, they're going to get more back in return. So how many of you are or have a PhD or are in the process of getting a PhD? So show of hands. So this is the etymology of doctor. Doctor is somebody who teaches. So we need to go out there and teach people. We need to go out there and tell people why developmental biology is important, why science is important. We had to literally go out there and grab people and tell them, right? Because, like normal people, otherwise, have no access to, this that we do in, here. So, there are many ways of doing this. There are the ones that give you a line on your CV, you TA or you teach. But we also have to embrace the ones that do not give you a line in your CV. And I know that they're less glamorous, but you actually have to go and make the effort to go out there and, visit the community and give back. Wrapping up, my plea for you today, be shameless about promoting developmental biology and promoting science. Follow, of course, people that you like in social media and beyond. And be kind to each other. Promote people that, you think that, you like, just, lift them up. I think that if everybody does this, once, imagine how many people are here in this room, and, how much good you can do with that. So in here today, my plea is, if you haven't done so, go and try to meet 2 or 3 people that you have never talked to here in this conference. Just talk about what you do, right? And what you have in common, especially what you do not have in common, right? Because the last plea from me is, once you have done that, then go out there and tell people why what you do is important, but also why the other 2 or 3 people you talk with that work in a completely different thing, why you think that's important. So if you're a developmental biologist doing mouse development, tell them why mouse development is important, but also why fly development is important. Because I think that community is the biggest middle finger that we can give to these people. If we are creating community every single day, and they cannot break that, then we win. Right? So with that, thank everybody who's played a part in my life. As advisors and mentors and friends and colleagues, everybody that I talk with, that I know in person, but I also that I do not know in person because we've all interacted through social media. So go out there. Tell everybody why developmental biology is amazing. Be shameful about it, and tell them that developmental biology is well and alive. So go give them help. Thank you very much.